Okay, you know that in paper one, there are some topics which you're supposed to cover. Please do not read those past, much past, like the previous trend before the curriculum was changed. So they will find out that some topics which are not supposed to be in the paper. But in this case, or for this year, these are the topics which you're supposed to have, and you're supposed to focus on them. Don't read on this topic and then you think that that is the I've killed that paper, I've killed that topic. You're only gonna get 8 marks. So for you to get at least the highest mark, make sure that response to environment by human, you get at least maybe a 40, get a 40 there maybe, and then here human reproduction, here also at least make sure that you get something like maybe 35 or 30, and then once you have these two, then now you can also add something if it's moving well. But this one and this one, there is a topic to pass. You can pass them easily. So you can also collect the marks there. If you see that this one is a problem, at least get a 25. Then you see that when you add them, you'll be able to get all the marks. So basically, those are the topics of paper one. So we're going to go through reproduction in vertebrates. We go through human reproduction. We go through response to the environment by human, response to environment by plants. And then after that, we will talk about the investigative question. And then we see if they ask you like this, how do you answer that? So we'll go through the investigative question, and then we see some graphical work. And then how do we mark that graphical work? Once you are done with that, then it means that you are ready to go, and you just have to put what we have studied or what we have covered in practice or in order. All right. Prepared use, you have to go through these biological terms, but I'll send them to you, and then you see how you're gonna check up yourself. So I'm not going to go too much time for this, but when you go to reproduction in vertebrates, when you come here and look at reproduction in vertebrates, this one, so we're gonna have to get these 80 marks. That is 5%. So it means that we're not wasting too much time in this. So what am I supposed to know when it comes to this? Let me just do like this. Fertilization. Here is just a concept which you just need to know that fertilization is when the sperm, you need to talk about the sperm, fuses with the ovum to form a zygote. It's a simple term, but it's better to say it's when the haplonucleus of the sperm fuses with the haplonucleus of the ovum to form a diploid zygote. That's basically fertilization. Or it's when the sperm fuses with the ovum to form a diploid zygote. That is also acceptable. So at least you must know that. If they give you a diagram, two different organisms, and ask you which one is external, which one is internal fertilization, you have to know at least. Or if they ask you some of the organisms where internal fertilization exists and where external fertilization exists, you must be able to identify. But you need to know that all organisms which develop inside the male's womb, then automatically that is internal fertilization, because it can't develop inside the male's womb and then after that it comes out and then it goes back. No. So everything which involves inside the mother's womb, it develops there, then automatically it is internal fertilization. So the only one which we can suspect to be external are those ones which develop outside the mother's womb. However, some of them are external fertilization, others are internal fertilization. For example, if you look at the chicken, the egg is laid, already fertilized. So what does that mean? It means that yes, the development is outside, but the fertilization is internal. But if you look at the frog, yes, the development is outside, but its fertilization is also external. So all organisms which have external development don't necessarily mean that it is external fertilization. So it can be internal fertilization, external development. So you have to differentiate between the two. Another thing which you need also to talk about when it comes to internal and external fertilization, you need to know that what are the advantages of internal fertilization over external, which one requires more sperms compared to the other. We see that external fertilization requires more sperm compared to the internal fertilization. Why? Because here the external sperms are not confined in a tube, while internal sperm is confined in the tube, so they move towards. If they get lost, not that much as when it's external. So you will see that internal has some advantage over external, or it has some features which make them to be favored compared to external. So you must know how to differentiate at least the two. If they bring the diagram, you must be able to score that mark. Then identify the characteristics of internal. Yeah, it's the same thing there. What are some of the advantages of internal over external fertilization? So if the development is, the fertilization is inside, so is in the tube, is within this tube, you deposit sperms here. It means that the sperms are going to go direct to the ovum. But now the ovum is here, sperms are deposited here. Some sperms are going to go there, some sperms are going to go there, some are going to go there, some are going to go there. So it means that only the sperm which has moved towards the ovum are the one which are going to fertilize the ovum. While in the tube, in the internal fertilization, you see when the sperms are deposited here, automatically they're going to go there. If they have some which go this side, but if this one is this side, this one is this side, we see that the chances of meeting the ovum is higher than when it is external. It's just deposited here. There's nothing surrounding it. Some goes there, some goes there, some goes there. Only these ones which go this side are the ones they are going to be what? Fertilizing the ovum. So we see that there is high chances of internal fertilization to have there is high chance for the ovum to be fertilized when it is internal compared to external. So when they say advantage of internal over external fertilization, we see that there are high chances of the sperm to meet the ovum and fertilize it. So there is a high chance of the sperm to meet the ovum so that successful fertilization can take place. So that at least you must know that. When they say that differentiate between oviparous and viviparous, here you have to know that ovi is ovum, of which the development is outside. Now this thing is like once you are done fertilization, there must be development. So when you say VV, in this case, let me clear up. In this case, we are meaning that development is inside VV. Yeah? We shall say that is what is inside. And then OV, it means the development is outside. So when this happens, it means that here the embryo obtains nutrients from the ovum, yes, which is outside the mother's body. Here, Vivi, the embryo obtains nutrients from the mother. How does it obtain nutrients? Through placenta. So here we need to talk about placenta. Here we need to talk about the ovum. Development is inside the mother's womb, and the embryo obtains nutrients from the mother through placenta. So we need to see that word placenta there. Here the development is outside the mother's body, and the embryo obtains nutrients from the egg yolk. So that is the major difference. So sometimes we can also add ovovivipary, of which here the development is inside vivi, but the embryo obtains nutrients from the egg yolk. Here the embryo obtains nutrients from the egg yolk, but the development is outside. Here the embryo obtains nutrients from egg yolk, but the development is inside. So those are the two major, those are the difference between ovovivi and then oviparous. Ovo, both ovo and the ovi, the embryo obtains nutrients from the egg yolk. But in ovoviviparous, the embryo is inside, or the embryo is inside, while in ovi, the embryo is outside. So we should not waste too much time there. At least when they bring the diagram, you must know, or you must differentiate between the two. Then we go to the next one, which is the advantages of viviparous and the ovoviviparous over viparous. Here, some people end up confusing this question with the question of development of, not development, with the question of external fertilization over internal fertilization, or internal fertilization over external fertilization. Here, you don't talk about sperms because we're not talking about development, fertilization. So you can't tell me about sperms here. Here we're talking about development. So when you have development inside, both of them, over the development which is outside, we shall see that the one which is outside, the chances of this embryo to develop, they are minimal because it's being exposed to predators. While the one which developed from inside, the chances of the embryo to develop are higher, because to get that baby which is developing, you have to first kill the mother, of which the mother is stronger, can also fight back. But when the development is outside, the mother might not be there. So it's easy for the prey, for the predator to feed on that prey. So here, the high chances of the embryo to develop. While here, no. Here the embryo is being protected by the mother as it is developing, meaning that maximum protection, of which here it is exposed to the environment. So when they bring the diagram and then we ask you some question related to that, you must know that in mind, that vivi and ovovivi, those ones, they are being protected by the parents. Oviparous, they are not protected by the parent. Then we go to the next question. They are saying that what is the function of amnion, chorion, yolk sac, and allantois? We call these amnion, chorion, yolk sac, and allantois extra embryonic membranes. So if they bring a terminology asking you about those, we are talking about, we call them extra embryonic membranes. Membranes which are extra to surrounding the embryo, meaning that the embryo has its own membranes, but we have other extra membranes which surrounding it. That's why we call it extra embryonic membranes. So now we need to know the function, at least you know one or two. One. Allantois is responsible for storage of wastes. I will not say it is, but I'll call it, it's a toilet. It's a toilet where wastes are being stored when the baby is developing. but you can't say it's a toilet in exam. Yeah, so it stores waste. I'm just giving you an example. Then yolk sac. Yolk sac, the function of yolk sac is a sac which contains, but this sac has connection to the baby. So it provides nutrients to the developing embryo. Then you have the chorion. Chorion is responsible for respiration and let me say gaseous exchange. That's chorion. However, when it comes to human being, chorion is the outermost extra embryonic membrane. When it comes to vertebrates or humans, we will see it, and it might perform a different function compared to the chicken or the birds. Then we have amnion. Amnion is responsible for protection, keeping the internal environment or constant in terms of temperature of this embryo. So it brings about free movement of the baby. Is that fluid where the embryo is playing around. So amnion, we have seen it, that's very good for baby, protecting the baby, shock absorber, allows free movement of the embryo. Chorion, respiration, and then a gaseous exchange. And then we say yolk sac is responsible for storage, providing nutrients to the developing embryo. In most cases, these people, they call it yolk. Yolk stores that, and then yolk sac provides it. Yes. And then we are saying that you have the allantois responsible for wastes. So those four membrane, you must know at least the function, and we call them extra embryonic membrane. We go to the next one, whereby precocial versus atricial. When they say precocial, you must know that pre means prepared. Atricial means it means all depends. Altricial. It has an L there. Altricial is, it depends on the parents. So eyes open, able to walk, have down feathers, can defend themselves. Atricial means that eyes closed, your health, everything depends on the parent. So because of that, there are some advantages of this over this. Yeah, precocial, it means that it is born when it is aware of the environment. Is cautious. Is born when it is prepared to the environment. While atricial is born when it doesn't know. Meaning that if the mother is not there, atricial might not be able to develop. While precocial would develop because it's able to look for the food. It can defend itself. It just needs some small techniques which can help it to boost it, but it can do what? It can survive on its own. Then, when it comes to parental care, identifying the characteristics, we have seen them. And then when we talk about parental care, what is parental care? Parental care, major function is basically protection, and it allows the organisms to develop until they do what? Until they mature. So that is the parental care. So our 8 marks, you must get them from these questions, and I know that by that time you will be able to get those marks. So if you look at this question, they're asking a question. Study diagram and answer the question. Which combination they show atricial development of the organisms? If you look at this diagram, you see that these babies, they're able to walk, they have feathers, the eyes are open, the ducks, you see them. But if you look at these ones, these babies, they can't walk. They are helpless. No feathers. No what? Eyes closed. They can't even stand. So it means that this one looks to be. Number two, if you look at this one, they are waiting for the food from the mother. Meaning that they are being provided everything. They can't move. You see even that the eyes are still what? Are still closed. So we're going to say that even this one is attritional. So in this case, the answer is going to be 2 and 4. Is there 2? 2 and 4. So D looks to be the most correct answer. So I'm not going to go through too much, but I was just trying to show you a simple question which they can ask you, and then you are able to answer such questions. So you have to know how to apply that. We go to the next question now. We go to the next section. The next section is going to be, we are going to have to drill. I say that we want 41 marks. 41 marks. We have to get these 41 marks. So let's get them. The first question is asking copulation, human reproduction. Copulation. Copulation is basically the insertion, the action of the insertion, or is the insertion of the penis into the vagina. That is what you call copulation. And then they're saying pregnancy. This is just a period from a zygote, when the zygote has been implanted, yes, until death. So pregnancy, you become pregnant. We say that you are pregnant when implantation basically has taken place. If implantation has not yet taken place, we might not say that you are pregnant. So that period, we can say from layman's, we can say that is when the zygote has formed until giving birth. That's the period. Sometimes we call it gestation. Scientifically, we call it gestation period. So now they're asking you, we will see this here. Why are testes hung outside the male's body? Testes hang outside the male's body to provide optimum temperature for sperm production. It provides optimum temperature or to provide temperature which is favorable for the sperms to be produced. That temperature is 2 degrees less than the body temperature, or 2 degrees less than 37.5. Don't tell me that if they ask you the question that what is the temperature sperms are supposed to be produced from, then you tell me 2 to 3 degrees. If the diagram is showing the temperature, you have to be specific. If the diagram is not showing, or the passage is not showing, then you can talk about 2 to 3 degrees. But it's better to be specific and say 2 degrees or or you say 3 degrees less than the body temperature. So which hormone is responsible for secondary sexual characteristics? The hormone is where? You have to first identify yourself. Where? In a male is testosterone. And then in a female is estrogen. Yes. So from there we need to label. We need to label. Let me see something. Let me just wait. Let me just give you this. So before we go there, we need to know, please go when you know how to label this diagram. Please go when you know how to label this diagram. Should I test you? Okay, let's label.
No, sir, we can continue.
Okay, you need to know. All right. So we're saying that, you need to know that's fallopian tube, not oviduct. Fallopian tube. That's a fallopian tube. This looks to be endometrium, cervix, vagina, but we can also ask you functions of that, or what's the second name for that. We can call it birth canal. That's a birth canal. Then this one is ovary. You must know the function of each. Fallopian tube, site for fertilization. Endometrium, that is where implantation takes place. Vagina, that's where the sperms are being poured, or is a passage for the baby during birth. Then you have the cervix. This one helps in the pushing of the baby. It opens so that it's able to push the baby during giving birth. That is ovary, very good for production of hormones, estrogen and progesterone. Number 2 is also good for providing the ovum. It produces the ovum. The second function produces hormones, that is estrogen and progesterone. Then when you go to the male, we see that when you look at it, make sure that these are testes. This tube is called vas deferens. So I will call A vas deferens. When you go out, the first one, which looks like a leaf, is called semina vesica. Then the biggest, this one is a bladder. Looks to be a bladder. And then this one looks to be the prostate gland. They didn't put the Cowper's gland, supposed to be there, but they didn't put it. But then this one is a penis. And then this one is the testis. And then this one is the scrotum. So you must know the functions. This one transports sperms from epididymis to urethra. You see, it brings it up to urethra. This gland, and this gland, we call them accessory glands. The glands which are found in the male are called accessory glands. And now we have generalized them. They have the same what? They have the same function. If you're using distinction material book, you know what I'm talking about. Then you have the testes. Testes provide sperms, secretes sperms, and also secretes a hormone testosterone. Then scrotum, that is basically for protection and also contract and relax so that it pushes the testes up and down to regulate the what? The temperature. Sorry. Then when we go to this, produce the sperm. The sperm has a head containing what you call the acrosome. So it's the acrosome. It has the head containing what you call the nucleus. So that's the nucleus. It has the middle piece. Inside here, we have the mitochondrion. Then you have the tail. The tail is responsible. We're going to see. Sometimes you call it flagellum. So it depends on what you have memorized. So the tail is very good in providing the propulsion force. Propulsion force means the forward force. We call it the forward force. The forward force. Then we have the middle. This one middle piece has the mitochondria. Mitochondria is responsible for providing energy so that the sperm can be able to move. Then you have the nucleus. We never talk about the nucleus. This is what must come into your mind. That is inheritance. First of all, It contains information for inheritance and also controls the activities of the cell, because this is a sperm cell, it's a cell also. Then you have the acrosome. Acrosome looks like the teeth in your mouth. The acrosome looks like teeth in your mouth. So this one contains chemicals, enzymes. Those chemicals, they contain enzymes. Yeah, they have enzymes which digest the membrane of the ovum. Fertilization. So 3 questions can come here. Draw the sperm. Make sure you know how to draw. Don't draw something like this. You do like this. There is a way how you guys draw those diagrams. You'll get 0. You'll get a 0. If your teacher is lenient and gives you something, at the marking center, they won't do that. So you'll get a 0 with eyes. So we are saying,
when you draw, do you like draw all the structures, or you only include the ones that like need labeling?
you must draw and include all the structures and label. In most cases, when they say us draw, make sure that draw and label, give a caption. Because what you're looking for, a caption, is it a correct diagram? And then we want to see that what you have drawn, yes, it has at least 2 to 3 labels. So you have to label, even if they don't ask you to label, make sure that you label. You at least put 2 of them. And then give a caption, a title, and then also make sure that you have drawn something which is correct, the correct diagram. That's when you're going to be able to get all the marks. But if you just want to draw small things which are not be visible, we won't give you marks, Baba. So that's number 1. Number 2, they can ask you the adaptation of the sperm for fertilization. In this case, you look at the head has the acrosome, which contains the enzyme digesting the membrane of the ovum for fertilization. Number 2, you can also look at the nucleus, which contains genetic material for inheritance. That is fertilization. They can ask you adaptation of a sperm, adaptation of a sperm for locomotion. How is the sperm adapted to locomote, to move? First of all, you talk about the shape of the head is pointed. The sperm is pointed, produce friction. Number 2, you can talk about the mitochondria in the middle piece. This one provides energy still for movement of the sperm. Number 3, you talk about the tail, long, provide strong propulsion force for forward movement. So this and the shape of the head, that is locomotion, movement. Then chromosome and the nucleus, that is adaptation for fertilization. So you have to know those things. Then on this diagram, all these are just labeling. You have to know how to also label such. Placenta, you see these are blood vessels connecting the placenta, and then you see these are the uterus, and then you see here is a fluid. This fluid is responsible, is amniotic fluid. We saw the functions, temperature regulation. You can talk about free movement, prevents dehydration. So this is the embryo or the fetus. Then we see the outer membrane. I hope I'm seeing it very well, but it looks to be the outer membrane, which is I'm not seeing properly, but one of it, I think it's the same, it looks to be the same, but one is a chorion, the outer membrane, outer membrane which surrounds the embryo. Sometimes we ask you the outermost membrane or the outermost extra embryonic membrane. How do you call that? We call it chorion. Please don't call it choroid, because choroid is found in the eye. This is called the chorion. Then we have this one, which is the umbilical cord. It's the umbilical cord. So from there, you need to now to go to this one. You need also to I understand this concept. So this one is an ovary. So basically it's showing you an ovarian cycle. So when it comes to this, you need to know how these developments are happening and what produces what. So in this case, we see the Graafian follicle developing. Graafian follicles developing. So it's developing. So just a trend for the Graafian follicles. It's still immature. You have the mature Graafian follicle with the ovum inside. So D is going to be regarded as the ovum, and then the outside part is going to be regarded as the mature Graafian follicle. So process of ovulation takes place, and then you have the ovum, the ovary here. I said the ovum here, and then this process is called ovulation. We have to know that when the ovum has been released, it becomes empty. So the empty Graafian follicle is turned into corpus luteum. So now we have this side whereby there is shrinking of the corpus luteum, or the corpus luteum remains big. So it depends on what happened to this ovum. If the fertilization takes place on this ovum, then automatically the corpus luteum is going to remain big. But if this one is shed away with the blood during menstruation, or fertilization did not take place, then the corpus luteum is going to start shrinking, and then the cycle continues. But now, once I know the cycle, now I need to understand which kind of hormone controls what and which kind of hormone is being produced. So as this one is developing, there is a special hormone which helps that to do so. That is FSH, follicle stimulating hormone, a hormone that stimulates the development of the Graafian follicle. So this Graafian follicle is developing because of FSH. Once I know that, now this Graafian follicle is developing. Once it develops, it starts to produce a special hormone called estrogen. The estrogen will start to prepare the uterus for implantation because of the incoming ovum, which is going to come. If fertilization takes place, then at least it must find everything when it's ready. So the Graafian follicle starts to produce a special hormone called estrogen. As the number goes on day 14, the ovulation takes place. Usually on day 14, ovulation takes place. But there is a special hormone which is responsible for triggering this process of ovulation to take place. And that hormone is called LH. But you need to understand yourself. You need to understand this, that where is LH coming from? LH is coming from the brain. The part of the brain called pituitary gland or hypophysis. So hypophysis produce the LH, and the LH actually, when the level of estrogen in the blood increases, it goes through the blood, and then it goes to the hypophysis. Hypophysis is going to be stimulated to produce LH. The moment LH is being produced, then the LH is going to come to the blood and stimulates the ovulation to take place. Then the ovum is going to be released. The moment ovum is released, now this empty Graafian follicle is going to be stimulated to be converted into corpus luteum, still by this hormone called LH. Now, corpus luteum, the moment corpus luteum is there, then it means that now I'm going to start producing progesterone. Estrogen is going to what? Is going to decrease. The moment estrogen decreases, it means that now the brain won't be stimulated to produce LH. So the level of LH goes down and the level of progesterone goes up. And then if fertilization takes place, yes, this corpus luteum is going to remain big. And then the level of progesterone is going to be kept high. But if fertilization does not take place, the level of progesterone is gonna drop. Why? Because corpus luteum is shrinking. And then the cycle continues like that. In this thing I've discussed or I've talked about, I've highlighted a number of questions they can ask. Even the question they brought in June, whereby they were asking you how is, I think they are saying that how is the progesterone interconnected with the with LH somewhere, I don't remember the question properly, but it is also answered properly there, whereby when this one increases estrogen, produces estrogen, estrogen goes to the blood will stimulate the pituitary gland to produce LH. Now LH comes here, triggers ovulation. You see, the ovulation takes place, and also LH also stimulates the conversion of this endographene follicle to corpus luteum. Corpus luteum produce progesterone. The moment progesterone is being produced, now estrogen drops. So you see that it is a cycle. If fertilization takes place, the corpus luteum is going to remain big like this, is maintained. If fertilization doesn't take place, the corpus luteum is going to shrink, and then it takes this route, shrink, and then it continues like that. So if I ask you from this diagram, is this person fertilized? The answer is no. Why? Because corpus luteum is shrinking. Now we come back to the questions. Identify the cycle above. Ovarian cycle. Process B, we have said is ovulation. What is D? We have said is ovum or mature ovum. Which hormone produced by A? We have you see now we are able to answer because we have understood the concept. Hormone produced by A is estrogen. Which woman is this woman? Which hormone? Not woman. Which hormone is produced by C? We said is progesterone. What is C? What is C? We said is corpus luteum. Which hormone is responsible for development of A? A, meaning that this is FSH. You see? Then they are saying that which hormone is responsible for development of C? Where is C? We said is LH. They're saying what is the individual pregnant? We say it is no. The individual is not pregnant. Give a reason for your answer. Corpus luteum is shrinking. Done. The ovarian cycle, done. We don't ask you, I don't think they can even ask you any question more than that. So you have to get those marks. Then we go to them. Which hormone is responsible for stimulation of sperms? Now we go back. We said this testosterone. What is the general name given to this the seminal, vesical, prostate gland, and corpus gland? We said we call them accessory glands. Then which cells are responsible for production of testosterone? We call them cells of Leydig. Cells of Leydig. We are saying that, let me just show you that. So basically we are here. The accessory glands. So we talked about the accessory glands. The cells which are responsible for production of testosterone are called cells of Leydig. And then asking you, nope. They're asking you that which cells are responsible for providing nutrients to the developing cells? For them, Sertoli cells. For them, Sertoli cells or Sertoli cells. They're saying that which cell will the person, if the vas deferens is cut, would the person be able to impregnate a woman? The answer is no. If you cut the vas deferens, you can't produce the baby because the sperms won't be able to be transported to the ejaculatory duct or the urethra. If the vas deferens is cut, so we have done that. Give a reason for your answer. Will the person be able to spread HIV? Yes. Give a reason for your answer is because there are other fluids which are being produced. So like blood or even these other fluids. So those fluids might be contaminated with HIV. If fallopian tube is cut, will the person become pregnant? No. If it's one fallopian tube cut, the person will become pregnant, but the fertility is gonna become low. But if both of them are cut, then the person cannot become pregnant. Give a reason for your answer, so it depends. If the two of them are cut, then automatically fertilization can take place because the sperm cannot meet the ovum. If only one is cut, the other one can produce the ovum. So, yes. Give two male and female secondary sexual characteristics. These are characteristics which, we asked you this in June, characteristics which show that you are sexually mature. You look at the characteristics which you were not born with. You were not born with the breast. You were not born with the voice, that kind of voice. You were not born with the hair under the armpits. You were not born with the pubic hair. You were not born with menstruation. So all that. So it depends on whether it's a male or with a female. So which hormone is responsible for secondary sexual characteristics in male? That is testosterone. And then that one for female, that is estrogen. Estrogen is responsible for secondary sexual characteristics in female, while that one in male is testosterone. Describe the process of spermatogenesis. But what is spermatogenesis? It's the formation of sperms. What is oogenesis? It's the formation of ovum. And then spermatogenesis and oogenesis, when you combine them together, what do you form? You form what we call gametogenesis, because these are gametes. So you form what we call gametogenesis. Yes, because those are gametes. So now you need to know how to describe the process of spermatogenesis and oogenesis. Here, the answers are straightforward. For the case of sperms, you tell us under the influence of testosterone, the diploid cells in the seminiferous tube would undergo meiosis to form 4 haploid sperm cells. That's what we want. We don't want more than that. Other books talk about a lot. We want more than that, man. Just want that. Under the influence of testosterone, the diploid cell in the seminiferous tube will undergo meiosis to form 4 haploid sperm cells. That's it. That's the process of spermatogenesis. Not more than that. Yes. So don't bite your tongue looking for answers you can't get. And then oogenesis, then this one is also simple. Under the influence of FSH, the diploid cell in the ovary will undergo mitosis. In this case, you first talk about mitosis, which will form numerous follicles. A lot of follicles, a lot of eggs, small eggs. Only one will enlarge. Only one will enlarge and undergoes meiosis. Once it undergoes meiosis, then it forms 4 haploid cells. Only one survives. You form 4 of them. Yes, only one survive, meaning that this one is the one which is going to enlarge, and then this one is going to become polar bodies. So under the influence of FSH, the diploid cell in the ovary undergo mitosis to form numerous follicles. Only one inside, only one cell will enlarge to undergo meiosis. And then out of 4, only one will survive to form a haploid ovum, or only one will mature. So oogenesis, spermatogenesis, those 2 processes, you have to know them. And it must not be a problem, because these are simple things which you must, a process which does not change. A process is a process. The way how you eat food is the same way you eat it until you die. The process, you can't put the food in the nose. You always go into the mouth. Okay. Draw the structure of a sperm. We saw it, and we say that make sure that the adaptation you do them. How is the sperm adapted to function? We said it. Look at the part function. The head has the acrosome to digest the membrane of the acrosome, which contains the enzyme digesting the membrane of the ovum. It's not the acrosome to digest. The acrosome contains enzymes which digest the membrane of the ovum. Then how is the sperm adapted to locomotion? You talk about all tail, long, for strong propulsion force, middle piece, mitochondria. So you talk about all those characteristics we talked about when we are busy describing the sperm. How is the sperm adapted to locomotion? Here you talk about the shape of the head, pointed to reduce friction, the tail long to propulsion force. Then you talk about the middle piece which contains mitochondria for energy. How is the sperm adapted for fertilization? We saw it. We say that you look at the nucleus for genetic inheritance. It contains DNA, or it contains genetic information. And then you can also talk about the acrosome, which contains enzyme for fertilization. Label and give the function of each part. So we have labeled all those diagrams. Yes, we have labeled all these diagrams. So if you look at this one, okay, we'll finish that afterwards. What is the consequence when the sperm has no acrosome? Then you ask yourself, what is the function of acrosome? The consequence is that the sperm will not be able to digest the membrane of the ovum. Therefore, fertilization will not take place, because the function of the acrosome is contain enzymes which digest the membrane of the ovum. List 4 hormones that which are involved in the menstrual cycle and their function. So we have seen them: follicle stimulating hormone, luteinizing hormone, progesterone, and estrogen. So FSH, we saw it. The one which we didn't talk about too much is progesterone. Progesterone prepares the uterus for implant. Progesterone maintains the pregnancy. By making the endometrium, you have to use the word more thicker, more vascular, and more glandular. It helps it to become more thicker, more vascular, and more glandular. Yes? So, progesterone maintains pregnancy by making the endometrium more thicker, more vascular, and more glandular. This word, more, if it's not there, you won't get a mark. While estrogen prepares the uterus for implantation by making it thick, vascular, and glandular. It's just thick. Check. It's just thick. Thick, a bit thick, a bit thick, a bit vascular, you see blood vessels, and a bit glands. And this one becomes more glandular. No, more vascular. You see a lot of blood vessels, very strong. More, check the gland, more glandular. If you compare this and this, you see these are big, more glandular, and then more thick. You see, so that's why I say more thicker. It was already thick, but it becomes more thicker, more vascular. It's already vascular, but now it's more vascular, and more glandular. It was already glandular, but it becomes more glandular. That is the meaning for that. Then is the person we saw that one from the graph. Give reason for your answer. Okay, we come back here. Let's check this more graph. Let's check this for more graph. Is this person pregnant? If you look at the graph, we see that the person is not pregnant. We see that the person is not pregnant. The graph is going down. So it shows that the level of progesterone is dropping. And if you look at here, the corpuscle is shrinking. So the person did not become pregnant because corpuscle is shrinking and the level of progesterone is dropping. So we come back and ask you, where are we? From the graph, so we have seen it. What is the influence of increased level of progesterone after day? If the level of progesterone is increased after day 26, it means that it's going to inhibit the production of FSH. If it prevents the production of FSH, then it means that no new follicle will be formed and no fertilization can take place. So that's why when the person is pregnant, the level of progesterone is high so that it prevents the production of FSH. Otherwise, you're going to become pregnant 2 times. You're pregnant 6 months and you're pregnant 8 months. Why? Because of the level of progesterone, when it is high, it does not prevent the production of FSH. Then automatically it's going to happen like that. But the function of progesterone, one of the functions, apart from maintaining pregnancy, is it prevents the production of FSH. So what will happen after day 26? It will influence the production of FSH so that is very low. And then there will be no new ovum formed. What is copulation? We saw it. Insertion of a penis into a vagina. What is fertilization? We saw it. We said that when the haplonucleus of the sperm fuses with the haplonucleus of the ovum, form a diploid zygote. Explain the process of fertilization. When the sperm come into contact with the ovum, the acrosome release the enzymes which digest the membrane of the ovum. You see? And then the haplonucleus of the sperm will fuse the haplonucleus of the ovum, form a diploid zygote. So ovum, sperm, let me just say like that. When this acrosome releases the enzyme, it will eat this, and then the ovum, meaning that it will be open. So the nucleus of the sperm will fuse the nucleus of the ovum, and then fertilization will take place. So that is the process of fertilization. So what is the function of the receptor? So this one goes to the next section, which is we have to go for another 54 marks now. 54 marks. But before we go to the 54 marks, let's see, just have a simple question here. Let's see if there's any question we can answer. Check like this now. According to what we have discussed, there are so many questions, but you see now, I think you are able to label. What is this? We see is vas deferens. What is this? We see it is urethra. What is this? We see the testis. What is this? We see the prostate gland. What is this? We see is fallopian tube. What is this? We see is the uterus. What is this? Sometimes you can call it uterine cavity. And then what's this is ovary. What is this is a vagina. So you see that we have answered this before. So don't go to the paper without knowing the labeling of the diagram. Don't, because this paper one, what makes it to be easy and simple is because of the labels. There are so many questions which involve labeling. So make sure that you get those marks for labeling. Explain why. Identify the part labeled B and the C we have seen. Provide the function of H, that is the vagina, receives the semen during copulation. Number 2, you can also talk about is a passage for the baby during the process of what? Giving birth. That's why it's called birth canal. State to the function of the fluid surrounding D with, this one, it provides alkaline fluid which neutralizes the acidity of the vagina. But remember now, any function you give is correct on this. Remember now we generalize them. And then now they're saying that explain why is it necessary for part C to be kept outside the body. We say that this part C, what do we say? We say that provide optimum temperature for sperm production. That is 2 degrees less than 35.7 or less than the body temperature. So you see that you can answer any question when it comes in this way. You must be able to answer such question. So it means that now we are going to the next section, which is section question 3, which is 54 marks. So it means that now we have 49 marks now. Let's go to the next one. Okay. Here What's the receptor? Receptor is an organ which receives, detects the changes in the environment. So it receives, it's a receptionist. Effector, it brings about the effect after the reaction has been detected. Detectable change in the internal external is called a stimulus. It's the what? Stimulus. And then electrical signal generated by the receptor in response to the stimulus, we call it impulse. So we say that is what? Where was I? Electrical signal generated by the receptor in the response to the stimulus, we call it a what? An impulse. What is the function of the nervous system? This one is basically to coordinate so that you're able to protect yourself, protect your body, to respond. Those are the functions of the nervous system. You are not harmed. So how is the brain protected? Meninges, cerebrospinal fluid, and meninges, cerebrospinal fluid, and the cranium. Not skull. Don't use the word skull. We call it what? We call it cranium, not skull. Not skull. Yes. Cranium, not skull. Manages the three membranes and then the cerebrospinal fluid, thus protecting the brain. What is spinal cord? Meninges, cerebrospinal fluid, and vertebra. That is the spinal cord. So you must not confuse that. Functions, no, this is brain. Obviously, you have to know when it comes to the brain. Cerebrum, what's the function? Cerebellum, medulla oblongata, corpus callosum, and how is the spinal cord protected in this case? Let's just use this question. I think I have a question related to that. Oh, I also had a question related to that. Here they're asking you, female, okay, why is a female want to know that the eye is fertile? You look at this and then saying that describe the development changes in the fertilized ovum until implanted. We like also to ask you this. Yes, we like to ask you this. A zygote is formed which undergoes mitosis to form a ball of cell called morula, which undergo mitosis to form a hollow ball of cell called blastula, which forms the outer layer called chorion, which forms finger-like projection called chorionic villi. So we like to ask you that this, yeah, is here. Most important. After fertilization, the zygote undergoes mitosis to form a solid ball. You added the word solid here now. You add the word solid ball of cell called morula, which undergoes mitosis to form a hollow ball of cell called blastula, which forms the outer layer called chorionic villi, which form the outer layer called chorion, which forms finger-like projection called chorionic villi for implantation. We like to ask you that. Anyway, we go back to our part which we wanted. Our part which we wanted. Who is it? I think maybe I don't have any question. Yes, this one. Let's use this one. If I'm to label, I see that this one is the cranium. See, it's the skull. Surrounding skull is the cranium. And then B, where's B? These are the three membranes. This one, and that one. You don't need to know them. Chiamara, dioramara, and arachnoids. We don't need to know them. We just need to know meninges. So these are the meninges, the three membranes we talked about. Then what is this? Cerebellum. What is this? That is spinal cord. What is this? Medulla oblongata. What is this? Pituitary gland. What is this? Cerebrum. What's this? Corpus callosum. So you need to know the function of each. Give the letter name of the part that coordinates all voluntary movement. It coordinates all voluntary movement. Voluntary movement are being coordinated by cerebellum. And then memorize this phone number. That is memory, reasoning, and judgment. Higher thought processes, memory, reasoning, and judgment. And that's the function of G, which is cerebrum. Give the name and a part of the diagram, or on the diagram D. What is D? Spinal cord. H. Corpus callosum. Spinal cord is a center for reflex action. Corpus callosum, it allows communication between the two hemispheres. You see? Give the function of E. You ask yourself, where is E? E is here. So if this is E, this is medulla oblongata. Give the function of E. Medulla oblongata is responsible to control all involuntary actions. How is this brain protected? By cranium, meninges, and the cerebrospinal fluid, the fluid which is inside, protecting it. So at least you must also know how to highlight that. Explain what is involved in the peripheral nervous system. Peripheral nervous system basically is made up of spinal nerves and the cranial nerves. 12 pair of cranial nerve and 31 pairs of spinal nerves. And this is what I'm talking about. No, not there. Here. Yes. Peripheral nervous system. They form what called sympathetic and parasympathetic, which work antagonistically. Antagonistically. See, peripheral nervous system. There's 12 pair of cranial nerves, 31 pairs of spinal nerves, meaning that these are the spinal nerves, the one which come from the spinal cord. Or the one which come from the brain, we call them cranial, from the word cranium, cranial nerves. So which is divided into two, which is autonomic and somatic. The autonomic is also divided into two, sympathetic and parasympathetic, which work antagonistically. So here are some of the questions they can ask you. How do you call the muscles which work in the opposite way? We call it antagonistic. And then we can also ask you the type of nervous system which restores the heartbeat after emergence is called what? It's called the parasympathetic nervous system. So you have to know these things. And then they're saying that explain what is involved within that response for storing, you see, after is parasympathetic nervous system. Give two systems working together in the opposite way. Call it antagonistic. What's the difference between sympathetic and parasympathetic? This one brings about emergence, then this one makes that organ to go back to normal. So the structure of a neuron, before you go to the paper, you must know how to know the structure of the sperm, structure of the ovum, and the structure of the neuron. Those are the three diagrams we can bring. Structure of the sperm, structure of the ovum, and the structure of the neuron. You have to know those ones. So, what is the axon function? Axon transmits the impulse away from the cell body. Dendrite transmits the impulse toward the cell body. And what is the function of the sensory neuron, interneuron, and these ones you have to know when it comes to the reflex arc. So, if this is the neuron, this is what you're talking about. This is the dendrite, brings the impulse to the cell body. This is the cell body. Axon transmits the impulse away from the cell body. They know how to do this. Or interneuron or sensory neuron. But in most cases, we bring the motor neuron or we bring the sensory neuron. And the major difference between the two is because the sensory neuron, the cell body is along the neuron, while here is at the terminal. You see, it's at the terminal. It's not along the neuron. So while here, it's along the neuron. Yeah. Yes, these questions. So what is the sensory neuron? What's the function? Transmits impulse from the receptor to central nervous system. Interneuron transmits impulse from the sensory neuron to connector neuron, to motor neuron. Motor neuron transmits the impulse from the central nervous system to effector. You see, here is from the receptor to central nervous system. Here is connect the this, and then the other one is to transmit the impulse to away. So that is it. So in that case, we go to these questions which you need to know as I come back with a reflex arc diagram. So what is the reflex arc? Is a path traveled by the impulse during the reflex action. We are talking about this path traveled by the impulse. This path is what you call the reflex arc. Path traveled by the impulse during the reflex action. Then, what is the function of that? The function is to protect the body from further harm. That's the function of reflex action. So then, what's the reflex action? It's a rapid automatic response to the stimulus. It's a rapid automatic response to the stimulus. What is the function? Because it is rapid, therefore it protects the body from further harm. Okay, give 2 examples of reflex action. Blinking of the eye. When you blink the eye. When you withdraw the hand from the hot object. The knee jerk, when you step on a sharp object, before you think, you have already removed. Why? Because it's controlled by the spinal cord. We'll see that question, why is it the brain too slow? Why are we not involving it there? Because the brain is too slow to react. Therefore, it might lead to damaging of the body. So what's the function of reflex action? Yes. Then the synapse. A synapse allows impulse to move in one direction. So what is it? Is a junction between 2 or more neurons. It's a gap between 2 or more neurons. So that junction of the gap, or the gap between 2 or more neurons, is what called a synapse. So what will happen if the motor neuron is damaged? Therefore here, we're going to look at the reflex arc, the structure, and then we explain this. But this, we're going to look at it when we come back after our short break. So we explain it in detail so that we are able to push it up. So when it comes to the reflex arc, I always tell my students not to complicate, because there's sometimes when they ask you to talk about the transmission of the impulse, but you can't know the transmission of impulse if you don't remember properly. Usually say Mr. M. Why is it Mr. M and Mrs. W? Then they have a bow tie. Ish. I have a bow tie. What's happening in this? Okay, and then a samosa here, bow tie, and then 2 samosas here. Okay. Now, what happens is that, for example, if this is the sensory neuron, when it comes, it has to be with the cell body and then out. Where the cell body is located on the sensory neuron, it swells out. So we call it a ganglion. And then now we have the connection there, which is called the synapse. Then junction again, which called a synapse. Then the motor neuron moves out and then it goes to the muscles. You see? Now that's the transmission of the impulse. This pass is what you call the reflex path. And then you have to know how to label these parts: sensory neuron, receptor, then dorsal root, ganglion, ganglion, and then you have ventral root, you have the muscles. Yeah, so all that you are supposed to know that. Why am I going all this way? Because of these function, these two questions. They say that what will happen if motor neuron is damaged? So you come back here. If this is the motor neuron X, and then you break it here, what will happen? It means that this sensory neuron is working and sends the impulse to the central nervous system. So it means that now this person will be able to sense what is happening in the environment, but won't be able to react because the motor neuron is damaged. The impulse won't reach the what? The effector. Can this happen whereby when you are able to sense but you can't react? Yes. Take an example when you have a muscle pull. You are feeling the pain, but you cannot move the what? You cannot move the leg but you feel the pain. Yeah? You feel the pain. Sometimes when someone is paralyzed, you see that he wants to move the hand, but he cannot move the hand. The hand is locked. But the mind is there trying to do things, but it cannot do that. Why? Because the sensory neuron is working, could be working, but the motor neuron is not be able to work. So the person will be able to sense, to detect, but won't be able to react. And then when it comes to this, if the sensory is damaged, then it means that the person, if this one is damaged and this one is working, then it means that the person won't be able to sense, but the person will be able to react. The person won't be able to sense, but the person will be able to react. Take an example when someone is removing a tooth. The tooth is removed. When it's being removed, the doctor will ask you to open the mouth. You open the mouth, you are reacting. When they are removing the tooth, you won't be able to feel, meaning that you won't be able to feel, you won't be able to take the stimulus, but you will be able to react. You won't be able to feel that pain. It's the same way Panadol is working. It's using the same mechanism when it's trying to fix you or try to reduce the pain. So it takes away the stimulation of where the pain is coming from and then stops it. But you continue working normally. So meaning that the motor neuron are working, but the sensory neuron has been intercepted or interrupted. Then the next question is saying that describe the process of reflex action. It's this one which we have tried to explain here, that the impulse is detected by the receptor, sends via the sensory neuron, and then the sensory neuron enters the dorsal root of the spinal cord, goes to the central nervous system, whereby it's connected by the synapse or synaptic connection. The impulse is sent via the interneuron, then sent to the motor neuron via another synaptic connection. Then the motor neuron moves out of the spinal cord, via the ventral root. Then it goes to that's disturbing me now. And then it comes here. Then it comes here to the effector. You understand? So this root is what you call the reflex arc. And if you come back here, the same root, this root is the root we are talking about, is what you call the reflexive arc. Is the reflexive arc. So it's the same type of root or same kind we are talking about there. When they say that describe the reflex arc, it comes as 6 marks. In most cases, it comes as 6 marks here. So you must be able to know that. Write down the path traveled by the impulse during the reflex action. Here they're talking about the path from the receptor to the central sensory neuron, then connector neuron, then motor neuron, then effector. Just the path. Let me see if I wrote it here. The path. Path. Yeah, this is the path they are talking about. Receptor, the sensory neuron, connector neuron, motor neuron, effector. But in most cases, they want you to write only this. Sensory neuron, connector neuron, motor neuron. Sensory neuron, connector neuron, motor neuron. Because they are talking about the neuron. If they talk about the neuron, then that's what you're supposed to give when it comes to that. Then, label the parts of the eye. I've not yet reached there. Difference between Alzheimer's and multiple sclerosis. We like to ask you this. Basically, Alzheimer's disease is related to being forgetting everything, and is as a result of the neuron in the cerebrum being damaged. The neurons, the cerebral cortex basically is damaged, where memory, reasoning, and judgment is. So that's why you have problems when it comes to memory, when it comes to reasoning, and when it comes to judge. So you can't manage to do that. Then the multiple sclerosis is basically as a result of the myelin sheath starts to degenerate. The myelin sheath, the layer which protects the layer which protects the what? The layer which protects the neuron starts to degenerate. So we call it myelin sheath. So once the myelin sheath is degenerating, then automatically you're going to have to suffer from what called multiple sclerosis. When you come back here, you have such questions. You see that now we are able to answer those questions. What is this? It's a sensory neuron because of this ganglion. And then what is this? This is the interneuron. And then what is this? It's a synapse. And then what is this? This is a motor neuron. That is a motor neuron. So from there, you see that what's a reflex action or rapid automatic response to the stimulus. Which one is representing interneuron? You see now, answer is there already. Which one shows the sensory neuron? Which one shows the sensory neuron? Which one shows the sensory neuron? Still the answer is there. We have said it that sensory neuron here is A. And then they are saying step 2, significance of microscopic gap. D. Function of a synapse allows impulse to move in one direction. What's the function of a synapse? It allows impulse to move in one direction. Also, it prevents continuous stimulation of the neuron. Then we are asking you, state 2, we are done with that. Explain why is the brain not initially involved in the reflex action? Because it is slow. So it end up, the brain will end up, the reaction, if the reaction is slow, you'll end up hurting your body. So the brain is not involved in these processes because you want your body to be a little bit faster. Draw a diagram of the sensory neuron. That one I told you, you have to be knowing the sperm, the ovum, and the neuron. So you have to know that. Then from there, we go to the eye. You have to know how to label everything here. Everything on the eye. Give vision involving 2 eyes. We call this binocular. So we're gonna go to that paper and then we try to do the labeling of the eye. Then what is accommodation? Accommodation is ability of the eye to see near and far object. So I will come back to that. Let's first go back to the eye and then we check and we see. Let's find the question of the eye. Yes, this is the question, for example. So you have to label it. A is what? A is the sclera. B is the cornea. What is C is the pupil. What is D is the iris. What is H? H is the suspensory ligament. What is G? G is the choroid. What is F? F is the lens. And what is E? E is the retina. So you have to know each and everything when it comes to the eye. What is this bending? Is called the yellow spot. That's where the clearest image is formed. And what is this? That is called the blind spot. How do you call that fluid? Is vitreous humor. How do you call the infrared fluid, which is found there? We call it aqueous humor. So how do you call these neurons which sends the impulse? We call them what? We call them optic nerve. So now we need to understand the structural changes which happens when someone is viewing a near object or a far object, which we call the accommodation. And then you also see what is PRELIM mechanism. When it comes to accommodation, there are some few things which you need to understand and remember. Any object which is more than 6 meters, we regard that as a far object. Any object less than 6 meters, we call that one a nearby object. So when you see an object which is near, it looks to be big. And then when you see object which is far, it looks to be small. It might not be small, but it looks to be what? Small. So this eye has the same mechanism. If you are viewing an object which is near, which is far, let me start with the far, the lens is going to be small. If you are viewing an object which is near, the lens is going to be big. Why? Because the object which is near looks to be a little bit much more bigger. So what happens? This light is supposed to be refracted. Refracted when it comes as a big beam, supposed to be refracted more so that it's bent to the retina. That's why the lens is supposed to be big enough to bring this light to the retina. But now, object which is far, it looks to be very small. If it's small, it means that even the beam which is going to strike the lens is going to be smaller compared to the one which is near, which looks to be what? Bigger. So, big object, big lens. Big. When the object is near, you see it as big. So, big must go with big. Then the small object, you see it as small, even the lens must be small. Now, your question comes in, what makes it to be small? What makes the lens to be small? We don't call it small, we call it less convex, scientifically, or more convex, or more round, less round. So, we can say that this one is because of the muscles which is attached on it, which called a suspensory ligament. So, when they become taut, they are being pulled, then they pull the lens. While here, the lens, the suspensory ligament, they slacken. They become loosened. Even here, they become loosened. The moment they become loosened, while here they become taut. So if it is become loosened, then the lens is going to become big. And then now it means that the object can be focused onto the retina, meaning that now the lens is going to refract more. While here, the light rays are going to be refracted less because the beam is small and then the clear image is focused onto the retina. So in other words, you have to know one. If you know this, the other one you just replace. Nearby object, what happens? The lens becomes more convex. Okay, the suspensory ligaments slacken, but now you have the ciliary muscles here. The ciliary muscles, they work in opposite way with the suspensory ligament. So I'm gonna say ciliary muscles contract, suspensory ligament, they slacken. When they slacken, they loosen. The tension on the lens, they don't pull anything on the lens. The tension on the lens decreases, the lens becomes more convex. Yes, once it becomes more convex, refracting power increases, and then a clear image is focused onto the retina. The same story here, nearby object, small object. So suspensory ligament, they have a ciliary muscles here. So they work in opposite way. If suspensory ligament, they are taut, automatically this one is going to relax. Ciliary muscles relax, suspensory ligament become taut, the tension on the lens increases, the lens becomes less convex, the refracting power decreases, and then light is bent less to the retina, and then a clear image is focused onto the retina. So you see that the concept is the same. The concept is the same. You see, nearby object, ciliary muscles, what happened? Contract. Suspensory ligament, slacken. Don't say relax. You will cross it. The moment you say relax, cross, they slacken, they loosen. That's the meaning. They don't relax, they just loosen. Loosen or slacken, the tension on lens decreases, meaning they're not being pulled. The lens becomes more convex. Now the light rays are refracted more, they are bent more, meaning that when it comes like this, they are bent more because they come as a big beam. A clear image is focused onto the retina. And if you go for this one, it's just the opposite of what you have just said on the nearby object. So you have to know that they work in opposite way. Then when it comes to PRELIM mechanism, this one is the ability of the eye to view an object in dim and bright light. In this case, we have muscles which we have to talk about. In this case, we talk about these muscles. You don't talk about anything else. You only talk about those muscles. The muscles of the iris, yes, and then the space between the iris. The muscles of the iris are called radial muscles, and then the sacral muscles. They can either relax or contract. So when they contract, if radial muscles contract and the sacral muscles relax, what will happen? If radial muscles relax and the sacral muscles contract, what will happen? So you come with the concept that you have dim light and then you have the bright light. In dim, you mix things. Yes, when the light is off, you end up mixing things. If you work, you write in dim light or in darkness, you end up mixing things. So in dim light, R is going to go with the C, and then C is going to go with R. While in bright light, R is going to go with R, and then C is going to go with the C. So radial muscles contract, sacral muscles relax. The diameter of the pupil, in dim, you open your eyes widely, meaning the diameter of the pupil increase, and then now more light will enter the what? More light will enter the eye. While here, bright light, radial muscles, I don't mix them. R goes with R. Radial muscles relax, sacral muscles contract. The diameter of the pupil constrict. The diameter becomes much more smaller, and then less light will enter the what? Will enter the eye. So that is pupillary mechanism. You just need to know that in pupillary mechanism, you have muscles which move in a sacral way, and then we have the muscles which move in the radial way. We have the muscles which move in the sacral way, and then we have the muscles which move in the radial way. So those two are supposed to be understood in that what? In that case. So from there, we go to the next one, which we say, these kind of changes which happen when the eye is dim. Okay, we have seen that. What is short sightedness? Here is the ability of the eye to see objects which are near, but you won't be able to see objects which are far. And how do we treat it? If you're able to see objects which are near, if you're able to see objects which are near, and then you have to first identify what caused that. What caused the object to be which are near. If this is the condition, we see that the eyeball is too long. You're able to see objects which are near, but you can't see objects which are far. So many of the objects which are coming from far, the image is formed before the retina. The moment the image is formed before the retina, it means that it is not forming on the retina. So the eyeball is too long. It means that the light cannot reach the what? The retina. That's why they're saying the eyeball is too long. Or the cornea is much more curved. Yes, if they're much more curved. So how do we solve this? We have to put on a lens which is going to diverge the light so that as if it is coming from a nearby object, and it pushes this point to the retina. That's why here we say that we put what we call concave lenses. A diverging lens, or we call it concave lens. So glasses which you put on, they must be concave lenses. And then the long sightedness is caused by object is too short, meaning that the image is focused onto behind the retina. Therefore, our job is to bring them back so that it's focused onto the retina. Therefore, in that case, we put the convex lens. So the eyeball is too short, you understand? Or the cornea is less curved. So it means that put on the convex lens, brings the light back, and then you'll be able to focus here instead of focusing at the back. That's what it means when it comes to long-sightedness and how to treat it. Astigmatism, basically this one is you form multiple images, and then here you can put on the prescribed lenses. You can put on prescribed lenses, very common in old age. Then the cataracts, that's when the eye, the lens crystallizes. It becomes milky, it becomes opaque, you can't see anything, which we call the cataracts. You have ever seen this situation? So we call it cataracts. So what we do, we remove and we do surgery, and then we replace it with the synthetic lenses. That's how you can treat cataracts. So the eye, in most cases, people, when they see the questions about the eye, they get frustrated. The eye is simple. You just need to focus on this. When the question comes, you will see that you will smash the what? You will smash the questions. Then when you go to the hearing, this one is also not that bad. Hearing is, you need to know the hearing process, labeling of the ear, then the balance. So let me check and see if I have any question of the ear here. Don't have any question for the ear. Okay, let's just go here. Here in the, if you look at this diagram, they ask you to label. They ask you to label. Make sure that you label. Yeah, this must be the marks you cannot miss. Kina, tympanic membrane, incus, and then semicircular canals, and then cochlea, auditory nerve, and then eustachian tube, and then round window. So you see, it's round while this one is oval. So the three bones are called oscars. But in this case, they are not asking for the three bones. They only ask for the bone which is in the middle. So it's Marius, it's incus, and stapes. Miss. Marius, incus, stapes. Some people, they call it has hammer, anvil, and stirrup. But in most cases, we want to use the English name. It's better to use the scientific name. So what if you know that's what you remember in the exam? You can try. You can try, but you will get, they could give a mark, but it's better to be on the safe side and say Marius, incus, and stapes. Then once you know that, then you must know the function of each. Then after that, you'll be able to know what you call hearing. Kina, correct sound wave, and directs to the auditory canal. So I'm explaining hearing, and I'm explaining the parts of the function of the parts. Number 2, tympanic membrane. Auditory canal sends the vibration to tympanic membrane. Tympanic membrane vibrate, converts the sound wave into the vibration, causing the oscars to vibrate. Also, the oscars amplify the vibration. The last oscar, yes, causes the oval window to vibrate too. The vibration of oval window will create a pressure wave in the inner ear, and then the cochlea, the cells in the cochlea, which we call the organ of Corti, will be stimulated, sending the impulse to the brain, cerebrum, for interpretation, so that now you are able to hear via the auditory nerves. Then we have these parts we have not talked about. This one equalizes pressure on the either side of the tympanic membrane. When they bring the ear, there are some parts which you don't miss. When they bring the ear, there are some parts which you don't miss. So this one, we see that the function is equalizes pressure on the either side of this tympanic membrane. How? By allowing air saturation in and out. You understand? Yes. And then we have the round window. This one absorbs the excess pressure from the inner ear. Round window absorbs the excess pressure from the inner ear. So that is it. So when we go back here, we know that the hearing is done and labeling of all the parts of hearing. Then we want to the balance. When it comes to balance, use side mode of balance. And there is even a song at the created for balance. Stay on our YouTube channel. You can check balance process song by ThunderEDUC. You'll find it. Okay. Then they're saying that if you go to the next question, we're going to use the side model of balance. So, to remember this, you know the stimulus, the receptor, and the location. Then you say to me that speed and direction, that is crystalline. That is what? That is crystalline. And then you have crystalline, you have where is it found? Location is found in the ampulla of the semicircular canal. And then you have the position of the head. Which receptor does that? That is maculae. Where is it found? In the circulus and the utriculus. The stimuli which you have detected is going to be converted into a nerve impulse, which is sent along the auditory nerve. Is sent along the auditory nerve for interpretation to restore the balance. So, when you are writing, how do I write? I'll write by saying that change in the speed and direction of the head will stimulate, change in the speed and direction of the head will stimulate the crystalli found in the ampulla of the semicircular canal, comma, change in the position of the head will stimulate the maculae found in the circulus and utriculus of the semicircular canal, comma, the stimuli are converted into a nerve impulse, which are sent along the auditory nerve to cerebellum for interpretation to restore the balance. So that is balance. That's the process of what? That's the process of what? Process of balance. So when it comes back here, you must know the two. These are processes which you can't change. It's supposed to be like that. You have to memorize that. There's nothing I can do for you for that. You just have to identify that method. Then what is middle ear infection? This accumulation of a fluid caused by bacteria in the middle ear. Yes, it causes that pus to accumulate. And how do we treat it? We treat it by apply antibiotics or grommets. So if you look at here, the antibiotics, these are grommets. This is the middle ear infection, is that side, is this side now. So what we do, we apply this grommet so that now it dries out this fluid. So that is it. Then we have how do you minimize the hearing loss? This one, you just remove the wax if there is caused by the wax. Or you can also do the hearing loss. You can also, if it is caused by a problem, maybe any tissue in the ear, then you can apply the hearing aid, or you can do what you call the cochlear implant if it's the cochlea which is being damaged. So you can remove the wax, which you call the cerumen, you can apply the hearing aid, or you can do what you call the cochlear implant. And once you know that, then it means that the ear is also done. There is nothing special or what hard making these things to be. There's nothing. Then from there, this topic, it means that now we have pushed them. We have pushed them. We have got this mark. We have got this mark. The moment you have these two, you are in almost, this is 5 and then this is 95. And you have also your 4 and these 8 marks. You will see that you're almost on 100. So look for 25 among this and this. Then automatically, once you have 125, you have a distinction. That is 80%. That's 80%. It's possible. It's possible. Distinction is possible. You just have to dedicate yourself, then read it. You see that these things are easy. Let's go to endocrine. Endocrine, this one is basically hormones. Hormones. I'm not teaching the real topic, but you need to know what are endocrine glands, exocrine glands, hormones, which hormone is that, and then the location. Endocrine is done. Then homeostasis, now you interact the hormones. And in this case, in most cases, I only explain one concept explain the other one. So it's the same thing, it's the same thing, it's the same thing. So hormones, these are chemical messengers which are produced in small amounts. They are being produced to the target organs. Sometimes they move through the what? Through the blood to target organs. And then in most cases, they are protein in what? Protein in nature. So when they ask you about the endocrine to exocrine, endocrine glands, they have ducts. Endocrine glands, they are ductless, while exocrine glands, they have ducts. So that's the major difference between endocrine and exocrine glands. The endocrine glands, they have ducts, while the exocrine gland, they have ducts, while the endocrine glands, they are ductless. This is what you see. The endocrine, they don't have ducts, while exocrine, they have this duct. A duct in science is called a tube. If you look at the gourd, some and the calabash, one is like that, and the one is like that. So this one is exocrine, or this one is endocrine. Then we ask you, give one gland which is both endocrine and exocrine. In this case, we talk about the pancreas. They ask you about that. Then label all the glands. So here you look at the gland and then you label all of them. You label all of them. You label all of them. In this case, we will just use this and then you try to label. Then you see how you have the pituitary gland, you have the hypothalamus, you have already mentioned it here. You have the thyroid gland, then here you have the adrenal gland, here you have the pancreas, then you have the ovary, and then you have the testes. If you forget, your eyes are the ovary, and then your head, your what is the eyes, ovary, and then you have the testes. And what is in between the testes? I don't know. So basically your face is the ovary and testes. Yeah. So you must remember now the eyes are ovary, and then the nose is what? Is testes. So you are able to remember that. All right. So you must know how to label, and then know what is the function of each gland you have talked about. Once you know the function, then automatically the exocrine and endocrine is done. What is the disorder characterized by lack of iodine in the body? We call that one goiter. Goiter. So that is the enlargement of the thyroid gland. That is enlargement of the thyroid gland. The enlargement of the thyroid gland, it is what you call the goiter. This one. So I think you have ever seen that. Then they are saying that what is hyperthyroidism? That is the same case as overworking of the thyroid gland. In most cases, when you don't have the iodine, you undergo that hyperthyroidism. The gland undergoes hyperthyroidism, meaning that it overworks, trying to produce, but it can't produce enough because you don't have enough iodine. And then a negative feedback mechanism. Now, these ones, you have to know all the negative feedback mechanism. I won't be specific here that this one is coming, but we like carbon dioxide because you have taken time. People don't like to read this carbon dioxide thing. Yeah, we like, in most cases, we like carbon dioxide. Yeah, let me not say that. Let me just keep quiet about it. But carbon dioxide, there are so many questions which related to glucose and the thyroxine, but in most cases they like carbon dioxide and then water. So you must know that, but use the same principle. Let me just give you the same principle here. But you have the normal level. You have the normal level of something. You can choose. We can say sugar. Sugar, that is glucose. Glucose. So you ask when that is high, then the last one is low. If this is low, one goes back to normal. If this is high level of glucose, so our aim is to see that we decrease this level of glucose. If you have here low level of glucose, our last level is to increase the level of glucose. You see, if you have high, this one must be decreased. If you have low, here must be increased. So now we have 2 points you're supposed to fill in here. The first one must be talking about the gland, and then the second one is the function of the hormone which is produced by the gland. So if you have too much glucose in the body, remember that glucose is converted to glycogen under the stimulation of insulin. So it means that I need to know the gland, which gland produces insulin, meaning that the pancreas here is going to produce insulin. Yes. So once I know that insulin has been produced, yes, the insulin, I'm going to talk about the function of this insulin. Insulin is going to stimulate the liver to convert. You see, I'm talking about the function now, function of insulin. Insulin does not convert. It stimulates the liver to convert glucose to glycogen. So, meaning that here glucose is converted to glycogen. Glycogen, you see? Stimulates the liver to convert glucose to glycogen. The moment glucose is converted to glycogen, automatically the level of glucose drops or decreases, and then it goes back to normal. It's the same story here. 2 points. Here is a gland, and then the function of the hormone. Function of hormone. So it means that if I have here glucose here, the amount of glucose, what's going to happen? The pancreas is going to produce what? It's going to produce glucagon. So yes, the glucagon, now what's the function of glucagon? The glucagon is going to stimulate the liver to convert glycogen back to glucose. You see, I'm talking about the function of glucagon, which is produced here. And then after that, the moment glycogen back to glucose, the level of glucose in the blood increases and then goes back to normal. So even if you use salt, water, any other, you will come back to the same point. You will know that if you have high here, it must be decrease here. Then tell me the gland, which hormone does it produce, and then you tell me the function of the hormone, and then the moment you tell me the function of the hormone here, then it automatically links here and goes back to normal. So when you come back here and you explain all this, it's the same. The only thing which does not click the same way is carbon dioxide. Carbon dioxide does not have the 2 ways. Yes, carbon dioxide doesn't have the 2 ways. It only has 1 way. Up here, what are the special cells responsible for production of glucagon and insulin? We call these isolates of Langerhans. Islet of Langerhans. Yes, easiness of Langerhans. Easiness of Langerhans. Those are special cells. We like to ask you that. Ne? And what will happen if the person level of insulin is low? You're gonna suffer from what you call diabetes mellitus. Diabetes mellitus. That is what you're gonna suffer from. And then other than glucagon, which other hormone result in the conversion of glucose to glycogen to glucose? Is adrenaline, because during emergence, glycogen is converted back to glucose. So the answer there is adrenaline. What is the hormone responsible for water regulation? That is ADH. Which structure in the skin responsible for controlling temperature? Which structure in the skin are responsible for controlling temperature? That is the sweat gland, and the blood capillaries. The capillaries, we're gonna see them. So let me stop here on temperature. Yes, on temperature, as I told you that they are closing now the building. So the structures in the skin which are responsible for controlling temperature, we say that it is the sweat gland and the blood capillaries. This is what you're talking about. These are the blood capillaries you're talking about, because the more they become nearer to the skin, the more heat they're gonna lose. Meaning that we will explain that. And then the sweat gland also is also involved in the structure controlling the heat of the body. Then, when we say that, which part of the brain is responsible for detecting the temperature changes? This one is the hypothalamus. Hypothalamus is the one which is responsible for emotion, hunger, sleep, temperature. So it is the hypothalamus. Describe the thermal regulation. When you say thermal, means heat. Thermal regulation during a hot day. What will happen during hot day? Remember, we said that the part which are responsible for controlling the temperature in the skin is the capillaries and the blood vessels, blood capillaries and the sweat gland. So in the blood or in the body, we have the sweat gland. This sweat gland is supposed to produce sweat. So this is called the sweat gland. It's called the sweat duct. It's called the sweat pore, and they produce what you call sweat. So sweat gland, sweat duct, sweat pore, sweat. And then we have the blood capillaries, which come like this. So this can be shrinked like that, or this one becomes bigger. And then we have the same story. That's big, while this is small, sharp. So in this case, we see that during hot, if this is the skin and this is the skin, during hot day, we see the blood vessels, those blood vessels outside the skin. Meaning that the capillaries, they look like this. So if they are looking like this, they are like this, then it means that more blood is passing through near the skin, and they are losing heat to the what? To the surrounding. And then this shunt is closed. That's why you see small here. While here, you see it's big. During cold day, more blood passes through down. That's why during cold, you don't sweat. You don't start sweat. And during hot day, you see that blood passes near the skin. That's why you are seeing yourself sweating. So what will happen if it's hot day? The hypothalamus is going to be stimulated, going to send the impulse to the blood vessels of the skin. the blood vessels are going to become dilated. And once they become dilated, more blood flows near the skin. If the blood flows near the skin, more heat is going to be lost to the surrounding. It doesn't stop there. More blood is going to be sent to the sweat gland. The sweat gland is going to become more active, produces more sweat, and then this sweat which is being produced is going to evaporate. As it evaporates with the heat, leading to the cooling of the body. So when we come to the exam, we see that the hypothalamus is stimulated. The moment your hypothalamus is stimulated, what will happen? It's going to send the impulse to the blood vessels of the what? Of the skin. The blood vessels will dilate, meaning that they become more wider. This is called vasodilation. You saying dilate is a mark, and this is called vasodilation is also another mark. More blood will flow near the skin, yes, and then more heat is going to be lost to the skin or from the skin. And then more blood is going to be sent to the sweat gland, and then the sweat gland is going to become more active. This being more active means that it's going to produce more sweat, and then this leads to evaporation of the sweat, will lead to cooling of the body. So we see that what happens during hot day is just the reverse of what happens during cold day. Hypothalamus stimulated, blood vessels of the skin is the impulse sent to the blood vessels of the skin. The blood vessels will constrict. It's called constriction. Less blood will flow near the skin, less heat is gonna. You see, the other one was more. Here is less. So it's just the opposite. Less blood is sent to the sweat gland. Less gland becomes less active. The less sweat is going to be released, and less evaporation, leading to less cooling of the what? Less cooling of the body. So you see that is just the opposite of the other one. So when they bring the question, for example, we see that here it is cold day because the blood vessels are constricted, while here is hot day because the blood vessels are dilated. And also this diagram shows it's a hot day because we see there is a symbol of sweat. So this means that it is possible, it is easy to sort this out. When they bring such question, make sure that at least you are able to answer those questions. So how will I differentiate between the blood vessels of individuals in the cold day and hot day? We have seen it here, and I've tried to explain them here. Hot day, they look like this, they dilate. Here, this one tries to be small, more blood flows near the skin. While here, the blood will flow down deep under the skin, and then less is gonna be sent. Near what? Near the skin. And if you look at this question, if you look at this, let me just go. You see that here the blood vessels have dilated. You see them, they are a little bit bigger, and then here the blood vessels are constricted. So if you check here, you also here you see they have dilated, and then here they have constricted, and then here you see that more there is sweat, and here there is no sweat. So this shows that this is cold day, while this one shows that is a hot day. And then from there, we go to the second last part, which is tropism. Tropism is a bending effect. Is when the plant grows towards or away from the stimulus. The stimulus can be light or short in this case. So give two function of auxins. Basically, these are growth promoters. So when they ask you about auxin, those are growth promoters. They promote growth. However, we shall see that they have different effects in the roots and in the stem. The effects are not the same. So you have to know what will happen in the root and what will happen in the stem. Which hormone is high concentration in winter? That is gibberellin, abscisic acid. because it calls all the properties it has. It is for winter. Seed dormancy, shading of leaves. We know that this one is winter. Well, if you look at the abscisic acid, gibberellin, it is summer. Why summer? Because of its functions it has. You see that gibberellins, it's very common in summer, is for germination of the seeds. You see, flowering. So that is, it happens in summer. So basically this is summer, this is winter, and then this is specifically for growth of roots and stems. Which hormone is high in summer? So we have said it. We also have said that due to they have this too much of what? Of germination. So if they bring a graph, just a graph like this, and then they say hormone, and then they say, and if this is winter, and then this is summer, and then they say this is A and this is B. Ask you which hormone is A and which hormone is B. We see that this hormone is low in winter but is high in summer. So it's gonna be gibberellin. And then this hormone is high in winter and low in summer. It's gonna be abscisic acid. You see? So give a reason for your answer, because abscisic acid is high in summer, in winter, due to dormancies and shedding of the leaves. So those properties of the plant, they happen towards winter or during winter itself. Then plant response to the stimulus in relation to light is referred to as light is photo. A response is tropism. That's the burning effect. So it's going to become phototropism. And then they ask you that which instrument is used to, explain the burning effect coming from a single direction. This one, they are talking about light. So here, let me just use this. Here we're gonna talk about what is that, what are the effect of the auxin. You need to know that auxins are growth promoters, in the shoot, found at the tip. Auxins are diffusible substance. Auxins, they diffuse downwards evenly, and auxins are sensitive to light. So if you know all that, then you come back. Auxins are found at the tip. Yes, when you expose light from a single direction, what is going to happen? If light is from this side, then it's going to move to this side. If light is from this side, then it's going to cause, remember we said they are sensitive to light. So these auxins are going to migrate to the dark side. And remember we said that auxins are growth promoters. So the dark side is going to grow more than the light side. Yes, leading to the bending of the shoot to the source of light. That's why here we're saying that when the shoot is exposed to unilateral light, yes, auxins move or diffuse to the dark side because they're sensitive to light. The dark side will have high concentration of auxins than the light side. The dark side will grow faster because auxins, they promote growth, and then the shoot will bend to the source of light. Simple. Then you have geotropism. Is a growth of a plant in a response to gravity or stimulus. When a plant root is, in this case, also you have to know the effects. That auxins are found in, they're found in their growth inhibitor. We say they're growth promoters. Auxins are found at the tip. It's the same thing. Auxins are diffusible substance. The same thing. Auxins move upward evenly. The other side, we said auxins, they move downward evenly. You see, downward evenly. So in this case, I will have to know. Once I get to know that, it means that if you put the shoot horizontally, the auxins are going to migrate downwards. The moment they migrate downwards, it means that the lower side is going to have much auxins than the upper side. If the upper side has more auxins, no, less oxygens than the lower side, then it means that now the shoot is gonna bend downwards. You see, it's gonna bend downwards. Why? Because the upper side grows faster than the lower side. Why? Because the lower side has too much oxygen in shoot. If it has too much oxygens, then it means that the oxygens, they're gonna inhibit growth. Remember we said that in roots, oxygens are growth inhibitor. So the side which has more oxygens is gonna grow less than the side which has more oxygens. It's gonna grow, the side which has more oxygens is gonna grow less than the side which does not have oxygens. While in shoot, the side which has more oxygen is gonna grow faster. Why? Because we say that oxygens are growth promoters. Those are the two key points you need to understand, that here the oxygens are growth promoters, while here the oxygens are growth inhibitors. The second point, oxygens, they fuse upward evenly, but now when you place it down, then they're gonna move downwards due to gravity. While here, oxygens, they move up or downwards evenly. But now if there is a sun, they're gonna move to the dark side because they're sensitive to light, and that will cause the changing. And then there is an instrument which is used to solve this problem of tropism, which we call a clinostat. This instrument helps in the breaking the effect of this oxygen. So when it's rotating, it means that it keeps giving this distribution equally. That's why here they're asking you that which instrument is used to remove the bending effect of oxygen. So it's called a clinostat. So it's called a clinostat. Then from there, just a moment. Then from there, you need to also understand how is the plant protected from herbivores. You can use stones, you can use chemicals also. Explain the bending effect of gravity. So we have seen that one. So there is a question, sometimes we bring it in the investigative question. I don't know whether they all these are different questions. Anyway, yeah, there is also this question, you see. In this case, you see that give the term of movement of plant in response to the stimulus. If you look at this, it shows that it's bending downwards due to gravity. So it means that it's going to become geotropism. Anyway, there is a question sometimes we ask you whereby we draw normal shoot, and then the shoot is cut. Then the shoot is cut, but we put a aluminum foil, and then a shoot is cut, but we put an agar plate there. Then we ask you A, B, C, and D. Then we start to ask you what will happen here, what will happen here, and what will happen here, and what will happen here after maybe 2 days. This one is gonna grow straight up. This one is gonna remain the same because oxygen, the tip is cut. So the oxygen is cut off. This one is gonna almost remain the same because the tip is cut, but they put aluminum foil which prevent the oxygen from going downwards evenly. And then this one is gonna grow. We have too much oxygen this side, so it means that it's gonna bend like that with the tip like that. So it depends on what they have given the question. You have to understand those two concepts of geotropism and the holotropism so that you don't get lost in that case. Lastly is about the investigative question. When you talk about the investigative question, you need to know the aim. Aim, whenever they ask you about the aim, it must start with the word to. Why did you do this? Is to. Why do you study? To. Why do you go to school? To. So it must start with the word to. And the aim, we can get it from the question. Or in most cases, with the question, when they give you a question, it has the aim. And that aim can help you to identify the dependent and the independent variable. So validity. Validity, it must start with the word same. What will make the result to be valid? Use the same equipment, same environment, temperature, same individuals, same, it must have same age. So it must be same. And then reliability, it must have a sample size. It must have repeating the experiment, but you have to apply. And this time you have to use the English. We mark the English. So if the sample size, how much they use? Maybe you use 5 individuals. They use 100 individuals. They repeated the experiment. So now, if the question comes and say that, how did they ensure? That is a past tense. So how did they ensure? So they used 5 individuals. How do they have ensured? They would have used more than 5 individuals. You see, so you have to reply according to the question. Don't just say, increase the sample size to what? 0. We'll give you a 0. So you have to apply when it comes to this. Planning stage, it must always have the word decide. Decide on the equipment, decide on the area, decide on the sample size, decide. It must start with the word decide. If you don't have the word decide, 0. Independent and dependent, in most cases, if they give you a graph, yes, in most cases, sometimes it's the first one is independent, the next one is dependent. Or go to the aim, the first statement, first or second. They'll show that the investigation has done to do this, to find out the effect of this on this. The effect becomes the independent, on this becomes the dependent. Then control variable is that thing which they did not change. It remained the same. And sometimes they ask you what is the function of that. Is basically to compare the results, to see that, yeah, the results are what? Are the same. So also sometimes we ask you to calculate the percentage increase and the percentage decrease. So percentage increase is always final minus initial. I just use a pen, not the fingers. Okay. Say that final minus initial over initial times 100. So if you started at 2000 and then you ended up in 500, so final is gonna be 500 minus 2000 divided by initial 2000. So the answer you get, it becomes that. But in most cases, since you said 2000 and then end up with 1000, so it's going to become percentage decrease. So it means that the moment you see that you have a negative, basically it becomes a percentage decrease. So, but the actual formula for percentage decrease is going to be initial minus final over initial times 100. So that you have 2000 minus 500 over 2000. So you see that the answer you get is the same, but this one has a negative value while this one has the positive value. So it shows that it is supposed to be percentage decrease. So you just swap this to this, but you have to know final minus initial over initial, that is percentage increase. Initial minus final over initial. Initial is always down. Times 100, it must be there. Final minus initial. The biggest minus smallest. If you forget anything, say big minus small over initial. So now find out what is the final. What is the biggest? The biggest in this case is initial, and the final is smaller. So it means that the initial, that's a percentage decrease. So you must know those concepts. And then the pie chart, please, when it comes to a pie chart, make sure that you use a protractor. Don't just use freehand. Use a protractor, and then there's a dot there. When you are busy drawing a circle with a compass, draw a line. After that, if they gave you 20, 40, 30, 10, and all these are percentages. All these are percentages. You can't plot this with the percentages. No, you have to first add them, plus 40, plus 30, plus 10. If they are percentages, they're going to make it out of 100. Then you tell me that 20 divided by 100 times 360, so that I'm able to convert them into degrees. 40 divided by 100 times 360. Then I convert them in degrees. Now use these ones, the degrees we have got, then start to measure on the protractor. Put a protractor there, put a line, measure. Maybe you say that is 75. Then from here, the protractor, you put it here now. You measure. Maybe you say that is 82. Then you protractor, put it here, measure. Then you tell me maybe 45. Put a protractor there, measure. So until the last segment. Put a caption and then put a key and then tell me that this means this. This means this. All those are marks. We give a mark there, we give a mark there, we give a mark there, we give a mark there. When it comes to the bar graph and histogram, it's not necessarily mean that you're supposed to start with a 0. Yes. And make sure that use a proper scale. If you start, you put a 0 here, then you need to tell us what is on x-axis and what is on y-axis. So if you do like this, then I'll know that, okay, if this is 0, this one can start with a 10. You understand? But if you put a 0 in the middle here, then we shall know that here is 0 and here is 0. And if we see that the range is not the same, even if these ones are the same, but the range here is not the same, you're going to lose the mark for the scale. So it does not necessarily mean that it is 0. So make sure that the point you want to start with here, it must be put at the first point. And also on the upper side, you must also put a point which you want to start with. When you draw the bar graph, make sure that the bars are the same size, same space, because all those things are, you end up losing marks. And then when you come here, you put units. You say days. Okay, say time in days. So you must put units. If you don't, gone. Put a caption. You don't put a caption. Caption must have both the dependent and the independent variable. You have only one factor, gone. The answer, the marks are gone. So easy to get the marks, easy to lose them. Small mistake, marks gone. So it means that you just have to be keen, and this question, they put them for you so that you are able to obtain at least a mark. Yeah, those people who are seeing that they are struggling with something, then you are able to get all the marks. Then lastly, on the line graph, histogram does not have any space. But line graph, we don't have a line graph which looks like this. We don't have a line graph which looks like this. We are not in maths. It can be like that, it can be like that, it can go up, it can do that, it can do that, but we don't have such. Yes. Once you see that, in most cases, as a result of you have swapped the what? The axis. And I think that is paper 1.